Hello again. In this lecture, we're going to speak about the development of the nervous system after birth, especially with a focus on the first few years right after birth, and we'll touch upon development as it continues to adolescence. The main focus here is on the structure of the brain itself. Of course, we'll be discussing some functions. However, in the next lecture, we will focus a lot on the environment, on experience, and then on the subsequent lectures of this course. We are going to focus on the specific systems, sensory and perceptual systems, as they develop. So today, we are going to go take an overview of postnatal development of the human nervous system with a special focus on the brain. The human nervous system, compared to other animals, it is a little bit slow, and it takes many years to basically reach maturation. And no sooner does it hit maturation than it starts to just go down into decline, unfortunately. So at birth, the baby, the newborn, is able to do some basic functions. For example, crying, swallowing, feeding, moving arms and legs. Basic functions, of survival to make the baby able to express his or her needs using crying and using, showing signs of discomfort and so on. So as a result, the nervous system is developed to that extent, and the baby comes in with some brain networks that are in default and in some kind of rough draft mode. And then the environment is going to shape more than two-thirds of the brain. Of course, it's not by quantity, but it's more about the connections, and the greatest, overwhelming number of connections and networks and circuits will be formed after birth. Now, if you compare this to an animal, say a deer, if you watch this video, I apologize if you find it a little bit gross, but the purpose is to show you in practical terms how an animal, once the animal is born, just within minutes of birth, you are going to see the animal standing up and walking and ready to within a couple of hours, basically ready to run and move around with the mother. So, of course, as a result, the pregnancy itself is prolonged, and the nervous system, it matures before birth. Otherwise, the animal is not going to survive. And this is the case with many, birds and, for example, birds. They come in the spring, and they lay their eggs. They take a few days, they hatch, and they're, within just a couple of weeks, they have wings, they're ready to fly. And they practice, and they're perfectly functional by the end of the spring and the beginning of the fall, when they start their migration. So think about it as how things are designed in terms of functionality. You want for example, a creature, a human being or an animal, to come at a certain point and to have certain functions. Then these certain functions, you will put in the brain and the nervous system the capacities, that will enable that animal to perform these functions. As a result, the development of the brain and the nervous system does not follow a consistent pattern of development. It depends on the areas and skills that need to be developed at a certain period in time, and based on that, you have parts of the brain that make this skill or this capacity possible. these areas in the brain develop first. So we're going to go over neurogenesis, synaptogenesis, apoptosis and synaptic pruning, myelination, genes and epigenetics, and the role of parenting. This is a big topic, but we will touch a little bit on it. In terms of neurogenesis, we understand neurogenesis is the production of new neurons, the birth of new neurons. So until 1997, it had been taken for granted that the brain wouldn't make any new brain cells. And as a result, they would cite evidence brain tumors are rare because the brain doesn't have the capacity to make any new cells. Once the neurons are born, that's it. Once they die and they are gone. However, in 1997-98, they discovered that in the hippocampus and a couple of areas around it, new neurons are born. And since that time, it has been well established so that there is a whole field of neurology called neurogenesis, focusing on the production of new neurons and how they function, how they work. For example, there are some neurons, as you are learning a new skill for the first time, these neurons are born instantaneously. Within minutes, they go and they get embedded. They go into a certain pathway and get out of the hippocampus, go to a certain pathway, and then go out and go back to the hippocampus, and then go back again so they repeat this a couple of 3 times or so. And then the third time, they're going to just go from the hippocampus and into the pathway and stay there for good. They spend the rest of their lives there. The neuroscientists see that as a way of connecting new information with old information as an interpretation. But the idea is neurogenesis is a process that lasts throughout the life cycle of a human being. And the particular area that is known to have that is the hippocampus and a couple of areas around the hippocampus. And the area in the hippocampus itself, which is the memory processing center that we discussed before, is the dentate gyrus. They call it dentate gyrus. It looks like it has teeth. And then the striatum, I'm going to discuss it afterwards, and that's it. So that is the kind of neurogenesis that lasts throughout life. For example, we lose brain cells every day, but in the hippocampus, the brain cells that are lost, they are replaced. And if someone has depression or has chronic stress, these epigenetic changes are going to cause suppression of genetic expression. So the genes in the hippocampus neurons will not copy DNA to produce new neurons. As a result, someone who has, clinical depression or chronic stress, that person could lose, the hippocampus could shrink by about 30%. Imagine your memory center shrinking and losing about 30% of its volume as a result of that epigenetic, that environmental kind of agent, which is stress or depression, that is going to cause to suppress genetic expression and prevent the neurogenesis in the hippocampus. When the person takes the medication and they go on the right track for treatment, fortunately, it regenerates again and the process is reversed. Of course, if the problem doesn't last extremely long. So recovery, of course, is going to depend on how extensive, how severe the damage, and how long, and so on. But that is just one example of neurogenesis, and without it, we will not be able to learn new information and forget and stuff like that. So neurogenesis, the production of new neurons, is a process that continues throughout life, but it is very in the hippocampus, and particularly, and another area, the striatum, which is, that part of the basal ganglia, these structures that are deep embedded in the white matter of the brain. And that's it. These are the only areas known to produce new neurons. However, we know that, of course, we discussed neurogenesis in early brain development and how it starts very early in the third week, and it continues to produce neurons at a very, very fast pace, and to populate the brain and the nervous system and so on. So that is a kind of different in that case, where you produce cells at a rate of 250,000 in 1 minute per minute, and then until you are able to produce 200 billion neurons. So, however, the majority, the overwhelming majority of these neurons now that are needed for constructing the brain prenatally before birth, they are basically produced by the seventh month. So at the end of the 6th month, a baby has a complete set of neurons. If the baby is born then, the baby can survive, and of course that will be premature, and but the baby physically will survive. The baby might need a lot of assistance, and there's a whole range of disabilities that are associated with preterm birth. So anyways, that process of neurogenesis is prenatally is very fast, very, very rapid, and it is synchronized with cell proliferation and cell migration spontaneously. Cells are born, they are shipped to their destination so that they can start to connect and to develop in the area where they are embedded. After birth, immediately right after birth, the baby, the neurogenesis targets one particular area, which is the olfactory bulb. So the olfactory bulb, as you could see here, that is some kind of basically the olfactory nerve that enables us to smell. And it goes in and on the base of the skull, above the nasal cavities, you have branches going in, and these branches line up the nasal cavities. So now you are able to smell. When you eat, the food gets crushed, and when you crush something, you produce heat. So you're actually heating the food while you are crushing it, and your body temperature is 98 or 97.6 to 98.6. So in addition to that, as you crush the food, you even heat it more, and it starts to evaporate, and then molecules from that are gonna go to your nasal cavity, and you're going to smell more. So as a result, more than 70-80% of taste is basically smell. So when we speak about smell, we speak about taste and smell because they're really connected. So anyways, that area here is the area where neurogenesis continues after birth, and cell migration continues so that it could populate this particular olfactory bulb, and it can make these extensions here so that they would be embedded and they start to function. Other than that, the other areas, the dentate gyrus and of the hippocampus and the striatum, they continue to have neurogenesis, but on a smaller scale, of course, and it continues throughout life, especially the hippocampus. So this is the dentate gyrus right here of the hippocampus, so this is one hippocampus here, and then it is kind of, you can see through it, and then you can see the dentate gyrus. And the striatum in the core of the brain. Last time in the previous lecture, we spoke about the basal ganglia. These are clusters of neurons and cell bodies that exist beneath, inside of the white matter in the core of the brain. And there are 5 sets of them, and 2 of them, which is that structure that is curving, is called the caudate nucleus. And another structure that is with it is the putamen, and they're actually connected, and the 2 together make a structure together are known as the striatum. So that is this area, these basal ganglia, as we discussed before, are extensively connected with the cortex, especially with the motor areas, with the prefrontal parts of the brain that are responsible for social communication, interpersonal skills, for emotional regulation, and also for language production. So the 3 major functions are based on 3 major pathways that go from the prefrontal cortex to the basal ganglia in terms of cognitive functions, emotions, and behavior, and like behaviors like language, for example. And they're of course adjacent to, they are near the hippocampus. Some of them are here, parts of them are here, and then the hippocampus is near them. So synaptogenesis is the process of forming synapses. Synapses we mentioned before. They can be formed between an axon of a neuron, like the axon comes in, and then it branches out into branches called synaptic terminals, each one. And the synaptic terminals, they extend, they connect onto the dendrites of another neuron. So the neuron that has synaptic terminals here connecting, that neuron is bringing in a signal. The neuron that has dendrites, and these dendrites receive the signal, because neurons can only send signals in one direction. Another way a synapse can be formed is to have a synaptic terminal connecting on the body of the cell directly, or even on the axon of the other neuron. So we're not going to get into these different kinds of synapses at this point, but what you see here is axons, as they kind of go to their site where they are supposed to stay there and function, whether it's an auditory neuron or a visual neuron or whatever it is. Once they go there and they are stimulated, of course they have molecular markers, agents, that are going to make them recognize their targets. And they have many thousands and thousands of neurons out there, and all of them are going to compete for the targets that they're going to connect on. And that competition is going to result in the best neurons are able to make the connections. And as a result, the ones that do not make connections, then they end up being discarded. So the neurons, when they start to extend, to grow, extend their axons, and then these axons have axon terminals, and then they kind of extend and they make contact, they form synapses with different kinds of cells. And once they have formed these synapses, then they start to kind of, with experience, these synapses become strengthened and protected. At the very same time, when the process of synaptic pruning occurs, it occurs spontaneously. You are dismantling something and strengthening the existing ones because they kind of are functional, they make connections. So synaptogenesis, the formation of new synapses, begins prenatally, and it is accelerated significantly, remarkably after birth, because most networks, most circuits develop and are refined after birth. So as a result, this refinement and kind of sculpting of these neurons based on the environmental experiences that you get, that is going to determine connectivity, how these neurons will connect. So that kind of pattern of synaptic connectivity is more shaped significantly by your experiences, by the activities that you engage the newborn in, and the nature and level and the extent of the experience or activity that the baby is engaged in. So every single hour of the life of that newborn is a precious time to interact, to stimulate the various sensory mechanisms of that newborn so that these connections can be made appropriately. And of course, we know when we will spend a lot of time speaking about connectivity and the effect of sensory deprivation or neglect and so on. So, synaptogenesis is, again, the formation of new synapses, and it does not progress like in a systematic way where you go and say, okay, this part is going to develop first and then you keep going back in a systematic way. It doesn't go this way. It goes based on what areas of the brain are needed at what stage to do what function, to do what purpose. And based on that, for example, you want a baby to even move before being born. So you need to develop the mechanism that will make that baby move, and and the mother's pregnant woman can feel the baby moving. I think 3 or 4 months. So as a result, to do that, you have to have an area that has connectivity strong enough to enable a movement of a limb. It is not an easy process. So as a result, the motor cortex is one of the earliest areas to be connected. The sensory cortex is also some evidence there is to say that the sensory cortex develops a little bit slightly before the motor cortex in terms of connectivity. So again, synaptogenesis does not follow a sequential pattern. It depends on the functions, on the specific areas that are needed to facilitate particular functions at a particular developmental stage. The cortex was mapped out by a scientist, a neuropathologist, way back at the beginning of the century. So his last name is Brodmann, and he mapped out the cortex into several areas, 52 or so, and based on the thickness, based on the topography of the cortex, based on what cells live in there and the cortical density and so on. And these are called, Brodmann area number 1, 2, 3, and so on. Of course, there are calls to move away from this, but that is not the point in our course. So when you hear about Brodmann area 41, for example, 41 and 42, that is the primary auditory cortex that takes signals as they come in, and it processes them, recognizes them as human speech or as the sound of a car or a cat and so on, and associates them with meaning and all of that. So cortical density, part of it is, a factor of it is synaptic density. When you have synapses, many synapses connecting, and the synapses are well developed, then in particular areas, that is going to contribute to the volume of the brain and to the density and of that part of the cortex. So synaptic density varies from area to area because, for example, synaptic density and the formation of synapses is going to depend on the availability of dendrites and of synaptic spines, little budding areas on the dendrites called synaptic spines, that the richer a dendrite is in synaptic spines, the more efficient it is, the more connections it can make, because synaptic spines are the points on the dendritic branch where axons of other neurons can come in and connect. So the greater the number of synaptic spines and the better developed they are, the more connections will that neuron in general make. So it creates like a whole mesh, and it makes that part of the cortex that is well connected and has a lot of synaptic density. It makes it quite rich and robust. There, as examples of how synaptic density changes from area to area depending on what part of the brain develops first and what part of the brain generally, has what characteristics. So, for example, there are greater synaptic connections and dendria in the frontal part of the brain. That is the part in the prefrontal cortex that enables you to perform self-regulation skills. Anything that you want to decide to manage your entire life, your entire, body, any decision that you make, your working memory, interactions, honesty, all of that is in the prefrontal cortex. So it is the thing that is working and making decisions for the entire body on a regular basis. So it makes sense that you need to have it better connected. And it is in the back, for example, in the visual cortex, it isn't as densely populated with, dendria and so on. There are less there. So in the visual cortex, which is the very back of the brain, that is the area that is going to receive input from your eyes, and it has, almost every neuron is going to pick one little piece of the frame that you have, that image that you see, and which is like pixel. And then you put all of these together, millions and millions of them together, and then you get a complete picture. So the auditory cortex witnesses a peak in synaptic connectivity around between the third and fourth months. So the baby, as we know, I'm just trying to connect this with how the brain itself functions. And why is that important? What will happen as a result of that increased synaptic connectivity between between the third and fourth month? The baby is born with black and white vision. The baby is not born to see in color. And at the beginning, the baby's vision is foggy and kind of sees at at the length of your forearm, basically, between this and that. That is 8.5 to 12 inches based on the length of the arm. So when you hold the baby like this, actually it is the upper part of your arm that is that. The distance between your hand when you have the baby and your face. So then gradually, as the cells in the retina start to connect and to get stimulated with light and so on, between by the 4th month, the baby is able to see in a full range of colors. And now you could see why this is possible at this time, because there's a big peak of synaptic connectivity at that particular area. In the motor cortex, for example, there's a peak at age 8 months, and that is going to get the baby ready, in a few weeks to start to talk, to start to connect what he or she has cognitively with the motor system and to produce meaningful speech. The visual cortex reaches maturation anywhere between the 4th month and the 12th month. And that big margin, of course, shows us the wide diversity in skills among the individuals. Some people have better skills developed, some people have middle level, some people have very high. And you could also understand that this margin, this great diversity, you have a lot of room to stimulate it with visual stimulation and toys and things and colors and, all kinds of things to accelerate, that connectivity. The auditory cortex, which is right above your ears, would be here. This is the temporal cortex. See, this black line is the fissure that separates the temporal cortex from the frontal and from the parietal. So the parietal cortex is for your sensations. You get, when you hold something, if you close your eyes and hold this and touch it, this is how you are able to feel this and then say, oh, I have a penny, I have a nickel, I have a paper clip because of this area here. That is your sensory. It has a sensory map of your body. We're going to discuss in a minute. The frontal cortex, the frontal lobe, right in front of this line that's called the central sulcus. In front of it, here, there's the motor cortex. And it has neurons that send their axons way down to go through the brain stem and spinal cord, and they are able to make movement possible. And again, we'll look at the motor homunculus and how it works. But the visual cortex, the auditory cortex, here in the temporal lobe, you have 3 gyri. The top one next to that line, next to the temporal fissure, the top one is called the superior temporal cortex. Right in the middle here, there is the primary auditory cortex that's connected to the ears, and this is what the area that receives auditory signals, and then it enables you to understand, recognize these signals and to kind of assemble them together. And there are some other areas it works with here. So this area is also connected with the visual cortex as well, and the memory center. So this area, the auditory cortex, begins to connect, and it witnesses a spurt around 4 months. And then it continues to also follow a kind of a similar pattern to the visual cortex. So in terms of the developmental foundations of the basic structures. However, it will take until age 11 years to 12 years for the top layers of that cortex to connect deeply with lower structures and to establish connections horizontally and in different areas. And 11 to 12 years is the age at which a child masters language, and the language of a child who is 11 to 12 years of age is very similar to that of the adult. So language foundations, language fundamentals and basics, basically language matures by age 11 to 12, because the primary auditory cortex and the structures that support it in the cortex are here. They mature at that time. So they facilitate processing and all kinds of language functions. We mentioned before, we spoke about how the cochlea itself and the inner ear and the middle ear, they reach maturation by before birth, week 20, which is like 5 months before birth. However, the cortex isn't well developed then, and is not very, is not yet connected very well to the. So you receive signals and so on, but you can't break them down. You cannot necessarily analyze or synthesize and so on. The processes of apoptosis and synaptic pruning, they start slightly prenatally. We said that by the 7th month, all the cells are born and a lot of connections are made. However, you need now to start to kind of get rid of the synapses that are not necessary, that are not connected well, that are deficient, and also of the cells, the extra cells that you don't need, that are no longer part of the network building process. So apoptosis is spontaneous cell death, and the cell has mechanisms. Up to 2010, there were more than 250. It was a sequence, a map of apoptosis, the mechanism inside of the cell that triggers apoptosis. And one triggers this, triggers that, it's controlled by that. It's a very, very complex map that it shows the programming of cell, programmed cell death. And what happens if that is figured, once that is figured out completely, and they know what is triggering, what and how the whole circuit works in terms of apoptosis, then cancer will no longer be a disease that we will know today. Once they have figured out that map of apoptosis, then they would have figured the cure for cancer. So, apoptosis, the purpose of it during brain development is to get rid of neurons that are no longer part of the network, that are not doing anything at all. And as I explained before, when you have a neuron that is connecting, you need to give that neuron space around it and in all different 3 dimensions so that it can grow longer, and it can grow more synaptic terminals, and it can grow more dendritic branches and spines. But if it is all kind of surrounded and has a lot of crowding around it, it will never have a chance to do that maturation. In addition, one of the aspects of the efficiency of neurons is the width of the axon. So, and the width of the synaptic terminals. And of course, again, if you do not uproot the excess neurons and excess synaptic branches and so on, then you are not going to have enough room to have the other ones, to allow the other ones to connect better and to develop and grow to become thicker and more effective. So even though apoptosis and then a nervous system begins slightly before birth, it continues mostly after birth, and it intensifies. And while it is spontaneously, the two aspects are happening together. While you are uprooting, taking out these unneeded neurons, you are constructing and building and strengthening and protecting the neurons that are already connected, that are needed for that work. It is spontaneous. You take this so that you can continue to build in its place. So by puberty, 50 to 70% of the neurons the baby was born with now are gone, are eliminated. And synaptic pruning, where you have, for example, the dendrites here, very extensive branches, and they have extensive connections. The nervous system kind of evaluates every single connection. If there's a connection that is dysfunctional, that is underutilized, that is not connecting, it will dismantle it so that it will redirect the energy to the branches that are connected and that are used. And if you ask anyone who knows about gardening, they will tell you that if you want, for example, a tree or a bush to grow horizontally, to be bushy, you trim it from the top. If you want it to become longer, you trim branches, lower branches of it, and branches on the side. Then it will put its energy to make the tree grow taller. So the process of synaptic pruning results in loss of about 40 to 50% of the synapses that were formed, that were originally formed. So by puberty, all of this excess dendrites and synaptic branches, they are gone, and also these neurons that are not needed, they are also gone. And the purpose is to allow more space to direct the resources that the nervous system has, that the brain has, to the neurons that are actually needed. Just to give you a quick idea about how this situation occurs. When there's a neuron, say connecting, this is a synaptic terminal, connecting with a dendrite. As long as this this neuron that is bringing a signal continues to fire and communicate with the receiving neuron, then every time it fires, the synaptic connection becomes stronger and longer lasting. And as a result, that neuron, the synaptic, the dendrite that is connecting to this neuron, they kind of depend on each other. If one is not doing its job, if this dendrite is dysfunctional and somehow this neuron is severed, has a severed axon and will not be able to communicate with another neuron, then the body is going to have signals to show this other synaptic terminal that you can give me signals, but I don't have the capacity to send them on the other side. So this neuron's going to figure out, so what the heck, I'm just going to withdraw, and it will withdraw itself, and then gradually it will either find another target to connect with. If it doesn't find a nearby target, then it will be dismantled and its energy will be directed to growing another axon terminal or strengthening the other ones that already exist. So this is what happens when there's injury, for example, that the body is going to, of the cell is going to start to shrivel. And once these axons, the neurons, hundreds and thousands that are connecting with that neuron, understand that this neuron is not functional anymore, they all withdraw. Once they withdraw from that neuron now is going to shrivel, and then it will start to undergo cell death. And it's It's a very interesting, process. But to make a connection, to make a synaptic, to make a synapse continue to function, to continue to exist, it has to be stimulated. If it stays for a long time, not stimulated, it will, withdraw. The synapses, synaptic terminals will withdraw, and then the cell body will withdraw, and these connections will be gone for good. Genes, we understand, as we discussed before, that the baby, when the baby, as soon as conception occurs, you have the whole process is driven by genetic coding, where you have triggers and genes start to be expressed to produce cells, and these cells exactly have genetic coding to know where they are going, what kind of cell they will develop into, and so on. So a lot of this is determined by genetics. However, the connectivity and the process itself of the gene itself, it has mechanisms that allows it, the genetic code that allows it to be reshaped by the environment. So genes themselves have the ability, the capacity, again, to allow the environmental factors to modify them, to make them express themselves and, cause a certain characteristic in the organism, or to suppress them and prevent them from expressing themselves. So either way, inhibition or suppression of a particular genetic, factor. So in terms of I think, let me touch upon something here that before I kind of go on, upon the myelination process, and then we'll speak about genetics and epigenetics more extensively. The myelination process occurs, most of it primarily after birth. Some of it, of course, before birth. But when we say that the brain comes to life, or the newborn is born with about approximately 1/3 of the size and volume of the adult brain, which is, the adult brain is about 3 pounds. So what makes the difference in terms of the bulk and the size? It isn't the number of neurons, because we understand the baby will be losing neurons, but yet we are gaining more. We are gaining 2/3 more. And the myelination process, the formation of these segments of myelin on the neuron, on the axons, that is going to account for almost most of the volume that is gained after birth. And the myelination process is selective, and it occurs based on particular areas. It doesn't follow a particular system or a pattern where you go from front to back or so and so on. No, you go to particular areas that are myelinated first, so that the baby can perform particular skills, and that will enable the emergence of specific developmental milestones. So, myelination is a mark, or a kind of one of the indicators of brain development. When we say brain development, again here, we speak about the connectivity, how we can make connections. But these connections are not necessarily going to be as effective if you do not have myelin and, well-insulated axons in the white, that make up the white matter. Then a lot of the energy will be lost, and the signals will not travel, will not be efficient. There is a whole range of neurological disorders that are caused by myelin loss, myelin degeneration, like Huntington's disease, like ALS, a whole wide range. If you don't have well-developed myelin or there's degeneration of myelination, the person will have all kinds of movement disorders and motor problems. So myelination continues robustly postnatally after birth and continues to develop in a robust fashion. It continues through the third decade of life. And in some research, for example, in the prefrontal cortex here, this is the area that receives or that's myelinated last. And some research shows that in males, it matures by about age 36. In females, about age 34. So you could see now that the process really isn't that easy. Myelination first occurs in the sensory cortex, or called somatosensory cortex, and then immediately after, like we speak about days after, in the primary motor cortex. Let's see what these are. So here's the brain, and here is between the pink line and the blue line, there is a fissure called the central fissure, or the sulcus, central sulcus. And in front of it is a gyrus. That gyrus is called the precentral gyrus. And that is in terms of function, it is the motor map of the body. It's called the primary motor cortex. And then behind it, on the other side, there is that blue gyrus. This is called the primary somatosensory cortex. That has the sensation for the entire body. It is part of the parietal lobe, and it has a map of the entire sensory map of the entire body. So, for example, look here. If you open this is the area between the two. So the two hemispheres are like this, and you open them up like this. There's a longitudinal fissure that goes from the front all the way to the back, making the right hemisphere, left hemisphere. So you can imagine this area here. So it goes this way. And then it continues between the hemispheres. So this is the primary motor cortex, keeping going. And then when it goes between the hemispheres, it extends. And then next to it, the same way, the primary somatosensory cortex goes. And then in between the hemispheres, there's part of it. So it's like this. So imagine yourself sitting, dangling your feet between the two hemispheres. And you are sitting here. The area that is next to the toes here and between the two hemispheres, that is part of the primary motor cortex, that moves your toes. Each particular component has a toe that it's like a switch that actually activates that toe. And it has neurons that go all the way down, send axons that go all the way down to send the signal to move your toes. And then above it, you have the area that moves the foot itself. And then here, the ankle. And then here, the knee and the calf. And here, right on top, that is the torso. And then the thorax, the waist and the thorax and the shoulder and the chest and then the arm and then the hand. See how much the hand is. It takes about 1/3 of the cortex is dedicated to the hand because, it's a major structure that actually shapes brain connectivity. And then a huge, about 1/3 is dedicated to the lips, the mouth, the eyes, the face, the larynx, the neck, All of this is dedicated to speech and swallowing and all kinds of functions. So you could see now, when you have the cell bodies of neurons all around here, organized in columns, and then each one is going to send an axon down, and they make tracks. And these tracks together are bundled, and they go down together, and that goes to the brain stem and the spinal cord, and that is what enables you to open your mouth, to move your lips, to lift up your tongue, and do all kinds of motor activities based on that. So every little area is particularly connected to one particular muscular system that it is going to move. On the other side, the blue area that is called the sensory homunculus. Homunculus in Latin means little man. You have exactly an identical map, but this map here, it does not move things. It enables you to get sensation. For example, here, you stick your tongue out. Here, you get the feeling, my tongue is going right, my tongue is going left, it's going up and down, even if you are closing your eyes. You know the location of the tongue in space. I feel pain, I feel pressure, I feel this, I feel that. Whatever sensation you get, that is going to be processed in this particular area, right here across from the movement part of the tongue. The lips here, anything you do with them here, that is going to tell you how they are feeling at that time. If there's pressure, if there's any temperature, whatever it is, that is going to be sensation. So every single part here is receiving signals, sensory signals, from that particular part of the body, and it basically covers the entire body. So we understand now why these areas are needed. You want the baby to feel pain. You want the baby to feel pressure. You want the baby to feel, so that the baby can protect, itself. And by withdrawing or by crying or by doing something, or to feel, pleasure, soothing and all of this. And at the same time to be able to move various structures that will enable the baby, for example, to move the lips, to suckle, to feed, and, to wiggle the legs or arms and so on. So it makes sense that these areas are myelinated first. So disruption of the myelination process, again, is going to cause a wide range of abnormalities. And one of these is a cerebral palsy that is, it occurs as one of the problems that result from myelination disruption. So genes and epigenetics, the interaction between genes and the environment. I spoke about genes and how genes, these are the sequences of DNA, amino acids, and they code. Every sequence has instructions for giving your body a particular structure or organ or quality, like say blue eyes, brown eyes, blonde hair, black hair, and so on. So the system comes in, the neurological system, the nervous system, it begins formation by predetermined genetic sequences and genetic genetic organization. So then 2/3 of the brain form and are stimulated by epigenetic factors in the environment that are going to cause the nervous system and, as a matter of fact, the body to adapt to the environment that this body or that nervous system is found on, is born into. So epigenetic changes can result from nutrition. If there's like poor nutrition, that definitely is going to affect the construction. You need particular components to build the system, And if there's anything that is going to interfere with that, it will cause a problem in the construction. Activity or the experience. This is a big thing. This is about everything. When we say activity or experience, it could be an activity or an experience when you speak to the baby, for example, looking at the baby. That's an activity. The baby now has an experience, saw you, looked at your eyes, looked at your face moving, and whatever it is that you can name, that is an experience. Whether you speak, whether you play, whatever it is. So, what kind of experience and the level of intensity, the level of exposure to that experience is going to influence how the brain is going to connect, how many connections the brain is going to make. So, for example, if you have a 3-month-old and you read with that 3-month-old and you continue reading and having the baby interact and laugh and play, and you are going to have a baby who is sociable, who loves reading, who enjoys reading, who enjoys being with others, and who also is highly intelligent because every day you are stimulating the auditory system, the visual system, the emotional system, and various systems. So all the sensory channels, all the sensory mechanisms that we have, about 21 of them, and we know the famous 5, smell, taste, and so on. And we know proprioception, which is sensation of your body in space and your muscles and your organs, how they are in space. But we have 21. For example, depth of vision, that is one sense. The sense that enables you to hold something between your fingertips and determine that this thing is thin or thick, and you compare it to something else, is, which one is thinner. That is a sense. Another sense is you hold something in your hand, and then you have a sense of its weight. What enables you to kind of say, oh, this is about 1 pound or 2 pounds? That's another sense. So anyways, these sensory mechanisms we have are the channels by which we collect information about the world around us and about the world inside of us. And these channels, we get the sensory stimulation from them, and these signals that we get go to the brain, and the brain transforms them into perceptions. And these perceptions become the foundation of our cognition, and our cognition becomes the source. It becomes the blueprint of our speech, of our behavior, and how we emotionally interact and how we react and so on. So it is a sequence, and we understand now that the environment plays a critical role in the construction of the brain, in the connectivity of how the brain is going to connect. And if that influence is negative, the consequences, of course, will be bad. If the connectivity the experiences are positive and they are ongoing, that the baby can count on, then that is going to lead to great, great gains. And we'll speak more next week. We're going to speak about connectivity and the critical periods of development. And so prepare yourself for something that's very, very interesting next week and the week after. So the environment, again, it provides the stimulation that is needed so that these networks and circuits can connect and can function effectively. If there is not enough stimulation, if there is no stimulation, that will be sensory deprivation. And that sensory deprivation is going to cause that particular channel connected to the sense not to function normally. and the person will not have the chance to develop the skills. Moreover, there are the critical periods during which specific kind of stimulation must occur at a particular level, so that these particular mechanisms or structures can develop. And if that stimulation is not provided at that time, then the person might have problems that could last throughout life. Prenatal care is another example of epigenetic changes or agents that can cause or influence genetic expression. So nutrition, the kind of activity or experience, the environment itself, is the baby found in a clean environment, in an environment that is well lit up, that is it has it gives a chance for exploration, for viewing and observing others, for being with others. If what kind of parental care is the baby getting? All of these are environment-based factors, epigenetic factors that will cause genes to either be suppressed or to express themselves in creating behaviors or physiological functions or physical. For example, studies made on animals, with animals, monkeys and so on, they show that baby monkeys that are raised by a real mother and they're caressed, they are groomed, they are touched, and so on, they develop, perfectly normally. And then they have a baby monkey that develops and has like a dummy, kind of mother, and that dummy has a bottle connected to it, and the baby goes to feed and so on. And you just find it's very abnormal. The baby doesn't grow social skills. The baby doesn't basically has so many problems. So we are going to discuss next week more about parental care and the conditions, for example, how the kind and nature of parenting is going to enhance connectivity and how it is critical during the critical periods of development. So this kind of lecture wasn't particularly extensive, but we wanted to cover the basics of brain construction and the nervous system, and give highlights about what matures and how it matures, what nervous system maturation looks like and brain maturation looks like. And again, in the upcoming classes, we are going to speak more about components and discuss them one at a time, and cognitive skills, and discuss them one at a time, and see how each one develops, and the factors and agents that will make it develop in a positive way or in a negative way. So I do hope that you found this interesting, this topic, and we will be speaking about connectivity and environment in the next couple of weeks. Thank you.